On any given day, millions of us are going about our daily lives unaware of tiny ticking time bombs right inside our own DNA. Alzheimer's, cancer, stroke, diabetes. Many diseases do not strike at random, but are predetermined by genetic variations and are thus inherited. But to what extent, and how can scientists use genetic modeling to better predict risk factors for a given individual and help further personalized health care? Join us as we head to the University of California, Los Angeles to speak with Eleazar Eskin from the Department of Computer Science and Human Genetics.
In the last 10 years, scientists have developed technologies that can measure DNA very effectively. And using these technologies, they were able to collect DNA from thousands of individuals. For example, if they're interested in understanding what genetic variants are involved in diabetes, they would collect 1,000 individuals with diabetes and 1,000 individuals without diabetes. And what they would do is they would look at the genetic variation, which is measuring the DNA at every position in these individuals' genomes, and compare to see if there's any genetic variants that are more common among the diabetics versus the non-diabetics. This technique is called the Genome-Wide association study and really got going in 2007.
Unfortunately, the link between genetic markers and disease is not at all as straightforward as one would like. First of all, there is not just one single gene that is responsible for a certain disease, or even a physical trait as simple as height. So far, scientists have discovered hundreds of genes that are involved in determining the height of an individual. But even when taking all of the genes currently known to be linked to a certain trait into consideration, there is still a huge gap between known and predicted heritability. This gap is known as missing heritability.
Even when they added up all of those variants together, they fell far short of what they expected to find. You see, decades ago, geneticists used twins to quantify how much information is predictive of disease risk. There are two kinds of twins, and the idea is that if you take how correlated a disease or a trait is among those two kinds of twins, you can quantify how much information there is in the genetic variation that predicts the disease. And based on these estimates that they've had for decades, when they added up all of the genetic variation that contributes to traits from GWASs, it fell far short of what they expected from twins. And this is what they refer to as missing heritability.
What exactly accounts for the missing heritability and how to improve current computing models is research material for the future. Current studies suggest that the more variants are included in the analysis, the better the models perform, which opens up great challenges and opportunities for computer scientists.
Around 2010, a group from Australia used a different technique called mixed models, and what they did is they try to estimate the contribution of all the variants in the genome to try to explain the heritability. And what they found using this technique, they can account for about half of the known heritability with these traits, which is a substantial increase over the 5% that was discovered in genome-wide association studies at the time.
Find out more in the review article Discovering Genes Involved in Disease and the Mystery of Missing Heritability in the October 2015 issue of Communications of the ACM.